Maybe you've heard of using genetic evidence to solve crimes, or using a specific piece of DNA to clone a gene. But how is this possible? Well, it all involves a process known as gel electrophoresis. Gel electrophoresis is used to separate genetic material, such as DNA and RNA, into fragments so that each sample can be compared. First, the samples of genetic material are placed in wells or small indentations in the gel. To better visualize this process, imagine that each sample is a team of runners. To separate each genetic sample into fragments, an electric current is run through the gel. The end of the gel box with the wells is placed towards a cathode, which is negatively charged, while the opposite end points towards an anode, which is positively charged. Because genetic material is negatively charged, it is pulled out of the wells by the electric current. Think of each genetic sample as a team of runners racing each other to the finish line. Though they are a team, each runner moves at their own pace. Because smaller genetic fragments move faster as they experience less resistance in the gel, each sample becomes separated by size, just like faster runners pull ahead of the others. This separation happens to each sample, resulting in a different distribution of fragments for each, just like each running team would have a unique spread of finish times. Because each genetic sample is unique, gel electrophoresis has various uses across the fields of genetics. It can be used to check that a PCR, or polymerase chain reaction which makes copies of a DNA segment, was successful. It is also used to construct DNA profiles, which allow for identification of an individual by their DNA alone. This is especially useful for solving crimes, because genetic evidence can be linked to a suspect. DNA fingerprints can also be used in genetic tests that determine relations between individuals, especially between children and their parents. As the field of genetics continues to expand, you can be sure that gel electrophoresis will have even more applications and an even greater impact on the future world.